Thanks to those who could join us. Thank you, Daniel, for joining us today for this really exciting topic. I did kind of paraphrase it into Bionics, but I know that it's much, much bigger than that. And I'm excited that this is your chosen field of pursuit and that you're able to share with us today some of your research and things that we can be looking forward to and things that are happening currently in the field of vision preservation. So if you'd like to tell us a little bit about yourself and your work, we can go ahead and get started.
Sure. I'm going to pull up my slides. So I'm sharing screen now. Go into the beginning. Press that button. There we go. Okay. So thank you so much. It's good to give a presentation for the Greater Detroit Agency for the Blind and Visually Impaired. My name is Daniel Rathbun. I am the research director for the ophthalmology department at Henry Ford Hospital. And my personal research is focused on bionic vision, how we restore vision to people with vision loss using electronic implants. And so I'm going to give a slightly broader talk today. I'm going to address the whole world of vision restoration, but there will be more of an emphasis on bionic vision since that's my area of expertise. For those who can't see me, I am a middle-aged white man with a shaved head and a goatee and glasses and a blue shirt, and I wore a bow tie for you guys. So the first slide here, and I'll be referencing the slides, I'll try to describe them as I go along. The first slide shows cyborgs. On the left are 3 real-world human cyborgs wearing 3 different implants, and on the right is a famous cyborg, Geordi LaForge from Star Trek. And when I was a kid, this is the cyborg that got me excited about science. I said, you know what, when I grow up, I want to help make his visor a real thing. And luckily, I've been able to get working in that field. So I study how the eyeball talks to the brain and how we can reconstitute that conversation between an electronic and the brain. So just because I have to do this for other talks, I don't have any financial disclosures. I don't own stock, and I'm not going to make a lot of money off of bionic vision. So for a little bit of context, whenever we're talking about restoring vision or any sort of medical intervention, there's this arc of time that we have to travel to develop that technology. And it involves basic research, preclinical research, clinical trials, and then once the FDA has approved something, it still has a lot of evaluations after the fact. And all of that is to protect patients and to make sure that the medical solutions that are made available to patients are as safe and effective as possible. So basic research, and a key part of this icon that I have on the screen shows that out of 5,000 to 10,000 compounds, starting at the very beginning of that pipeline, over the course of that process, it gets whittled down to just one FDA-approved drug or treatment. So basic research, that's kind of where we develop the idea, and that can take many years, 5, 10 years easily. Preclinical research is where we do the animal tests to make sure that something is somewhat effective and that it's somewhat safe before we're going to start working with humans. Phase 1 is in the FDA clinical trial process is where we evaluate the safety of the intervention and how much of that intervention to give if there's a dosing that needs to be worked out. Phase 2 is efficacy. And that's where we evaluate in the best situation, best case scenario, does this treatment work? Does it do what it's supposed to do? Or at least, is it somewhat effective in doing what it's supposed to do? Phase 3, the last phase, doesn't always happen, but it's a question about effectiveness. And the distinction between efficacy and effectiveness is in the real world, when the doctors are actually implementing this intervention with real world people who aren't being monitored in a clinical trial, do we get improvement that's better than the current benchmark? This is where the companies will do their pre-marketing. And then once you get to market, there's post-marketing surveillance, which is sometimes called phase 4 trials. And so that's that whole process. And the reason I mention that is just to give you guys an idea of this long timeline from the origin of an idea to the actual availability of that idea on the market. And, you know, we've all been experiencing something very different in the last 2 years, in that we saw this idea for the COVID vaccines, which they were already somewhat through the basic research stage and a little bit of preclinical. We saw them just jump through that 5 or 10 years worth of clinical trials over the course of 12, 18 months. And so that's very abnormal. It's a great success story for modern medical science, but that's not how things normally go. A little bit more background before I dive into sight restoration strategies. I think most people with some sort of vision loss are probably somewhat familiar with the visual system, but I'll give a little overview anyway. So light comes in through the front of the eye, the cornea, which is clear. It passes through a lens in the eye, which is also clear. It passes through the goo in the eye, which is the vitreous, also clear. And then it passes through and interacts with a tissue called the retina. And the retina is this layer, very thin, wet tissue paper-like layer of neural cells in the back of the eye. And that is where photons of light get turned into electrical chemical signals from neurons. And so that's the retina. And then the retina has wires, axons, little pieces of the cells that come out of the eye through the optic nerve, and they go into the brain stem, into what's called the lateral geniculate nucleus of the thalamus. And that is kind of a relay that takes that information from the eye, does a little bit of processing, then sends it on up to visual cortex, which is on the surface of your brain, kind of buried in between the two halves of your brain at the back of your head. And so that's the basic anatomy. And blindness can occur with an interruption of that pathway of information anywhere along the line. If your cornea becomes scarred, then you can't see through it, and your whole visual system works, but light's not getting to your retina. If your retina's sick, then even though your visual brain may work, your retina can't send signals to the brain. Similarly, if the thalamus is damaged, you can lose information there. And the visual cortex can also be a source of vision loss. And it's important to understand the anatomy, or at least be somewhat familiar, because where the vision loss has occurred in that process determines what interventions are going to work or not work, or at least have a potential to work. And I'll skip this. That's a little icon showing the layers of the retina. So there's a lot of different interventions that have been tried. A lot of different ways people have tried to restore vision in vision loss. The first I'm going to talk about is gene therapy. But there's also cell therapies like stem cells. There's also neuroregeneration. And then bionic vision, the electronics interacting with the brain, is what I'll talk about in a little while. But let me take you through some of the background on these. So gene therapy has been around for 15 years or so as a viable option. There's been a few dozen trials since 2007, first one in humans. And the most successful has been Luxturna, which you may have heard about. And Luxturna was developed for Leber's congenital amaurosis, which is one of the inheritable forms of blindness where the retina degenerates. And it involves an adeno-associated virus that's been neutralized. So it's actually a naturally occurring virus where they take out all the virulent factors and they use that virus to carry genetic information into the cells of the body. And so that virus with the correct gene, a gene that's not broken, is injected into the eye. The virus gets into the cells of the eye, injects that gene, and that gene then gets translated into a protein. And so if you have a genetic defect, for example, in Leber's congenital amaurosis, that makes a bad protein in your eye that causes blindness, this can restore that protein and address the blindness. So importantly, Luxturna is the very first gene therapy ever approved, not just for blindness, but anywhere. And this is kind of a common theme in that vision tends to be where new technology comes around and finds its first practical applications. Artificial intelligence also has its first medical application approved by the FDA in vision. So it was the first gene therapy to be approved by the FDA in 2017. And as of March 2018, you can get Luxturna injections if you qualify, if you have LCA and other qualifying factors. And it only costs you $425,000 per eye. So for just under a cool $1 million, you can get each eye injected and have some vision restoration. I think the younger you are, and this is a disease that hits people right after birth, so it's good to try to get the intervention in early. But the younger you are, I think the better results you have. When this was first developed, the company that developed it actually took some blind dogs that had received this therapy into the halls of Congress and showed them to lawmakers, and the dogs were behaving like normal dogs, and everybody was wowed. It's, hey, they can see, and they could see better than they could without the injection. But critically, dogs don't need to see very well to be fully functional. I have a dog that has, actually, she was a leader dog pup, and she's blind in one eye, and you can't tell unless you know what you're looking for. Anyway, so dog vision's been restored pretty well, but when this Luxturna injection has been given to humans, the improvement is less good. And it's not, there is improvement, but it's not restoration of sight. So what the FDA reviewers said in 2017 when they were approving it was, number one, we don't expect this to restore normal human vision. Number two, only about half of the treated patients had improvement that met the minimum requirement to be able to say, yes, this person got better. These improvements might not persist. That gene that's injected into the cells, it can degrade over time. And so it's not clear yet whether follow-up injections are going to be needed in the future, although we are hopeful that they won't be. And there were 2 patients in this trial that experienced permanent vision loss. And if you've gone from low vision to ultra low vision to full blindness, you probably know that even a little bit of vision can give you a lot of results compared to no vision. So that's a serious concern. So that's kind of where things stand now. Luxturna is on the market. You can talk to your doc and get it if you have LCA and you qualify along other parameters. You know, you're not severely ill from other diseases, that sort of thing. But it's only for LCA, and that's one of the downsides of gene therapy is every single genetic defect has to have its own clinical trials, its own therapy developed, a different gene developed. And for inherited blindnesses, there are 300 genes we know of right now, each of which can cause blindness. So that's a lot of clinical trials and a lot of time. One thing that's coming down the pipeline you guys may have heard about is CRISPR-Cas, which is a different way of editing genes and may accelerate the preclinical side of things. There's another drug that was recently developed called Lumavoc, and this comes from GenSight Biologics. And this one, instead of LCA, it's for Leber's hereditary optic neuropathy, which is a mitochondrial disease. So it's something you can inherit from your mother. And so far, their trials have shown clinically meaningful improvement, so something you can actually detect scientifically for at least 4 years. So it persists, and there is detectable improvement. GenSight, which is based in France, got the authorization for temporary use in 2021. So that's kind of a special, it's like FDA approval, but provisional. And in January 2022, the FDA evaluated their work so far and said, you know what, it looks good, but we need more placebo-controlled work. And the main reason for that, I don't have the slide, the main reason for that is that it turns out there's bilateral effects. So patients who get the injection in their right eye, they see improvement with their right eye. They, I think it's like 15, 17 additional letters on the Snellen chart, on the reading letters chart. But they also got improvement in the eye that didn't get an injection. And that shouldn't happen. That's not supposed to happen. And so there's this concern of, well, what exactly is going on? We need to understand this better before we put it on the market. And those are the sorts of setbacks that every approach to sight restoration encounters along the way. So I'm not saying that the gene therapy is doomed to fail. In fact, I think it's going to be pretty successful for inherited blindnesses. But we've still got a long way to go before most genetic defects have a treatment, and those treatments bring people's vision back to more or less normal, full vision. So that's gene therapy. I'm going to move on now to optogenetics, which is another approach to restoring sight. It's also a gene therapy, but it's a very specific gene therapy. And what optogenetics involves, and I've got a little icon here, that's a little image that's showing the cell membrane. And what optogenetics involves is injecting genetic material that will create proteins that sit in that cell membrane and respond to light. So take a step back. Many forms of blindness, retinitis pigmentosa, many inherited retinal diseases, and macular degeneration involve the loss of function of photoreceptors, those cells that turn photons of light into a neural signal. So if you don't have any photoreceptors, your whole visual system can work, but you can't turn light into something your brain understands. Optogenetics involves making other cells, not sick cells, but other cells in your retina respond to light. And so one of the first approaches that was developed actually took a light-sensitive protein from algae, to do photosynthesis, and took this protein from algae and injected that in human cells and made them respond to light. So since about 2006, we've had over 20 trials, most of them done in mice. And each of them show some improvement. The kind of dirty secret of our field is mice don't have very good vision. So it's relatively easy to restore them to full vision, where you can't tell the difference between the treated mouse and the healthy normal mouse. But that's just an aside. So since 2006, there's been almost 20 trials in mice, preclinical trials. It's worth mentioning that Detroit hosts one of the key innovators in this field, Zhu Hua Pan. Dr. Pan at Wayne State University actually has been developing these proteins for years, and he's recognized as one of the critical leaders in this field. And he developed an approach, an optogenetic therapy that was bought up by a company called Retrosense, a little startup biotech company. And in 2016, the first human, a woman in Dallas, Texas, was injected with this in her eye and had some visual restoration. Later on, Allergan, this big biopharma company, bought up Retrosense. And the sight that was restored was disappointing. There was sight restoration, but it was very limited. And so essentially what people in the field like me can say is it looks like Allergan, they own that intellectual property, they've got that technology on the shelf, but they don't seem to be aggressively moving it to market. So that's where that stands. But they're not the only player in the world. So in October 2018, another human clinical trial started with a guy named Jose Alain Sahel, who is a huge name in French biomedical vision research. Working along with the GenSight Biologics company in London, they started a clinical trial with their own approach to optogenetics. Another company named Nanoscope developed the Crimson R optogene. And this one, they put that gene in, and it infects retinal ganglion cells, which are the cells that have wires that connect the eye with the brain. Their axons go from the retina all the way out to the brain. And this optogene makes those cells sensitive to light. And so they can respond to light and then send the signal directly into the brain. Unfortunately, that gene, or that optogenetic protein, is not very responsive to light. It's not very sensitive. And so what they have to do is they have to have a goggle system that has a camera, takes a movie of the world, and then it processes that image to make it sharper and higher contrast, and then make it really bright in just the right wavelength of light so that these cells will respond to the light. And there was a report that came out last year in May with 11 patients who reported that statistically significant showed that they could detect objects They could locate where they were. They could count them. They could reach out and touch them without making errors. And more importantly, this only works if your eye is injected and you have the goggles turned on. So if your eye is injected but the goggles are off, it doesn't work. If your eye is not injected and the goggles are turned on, it doesn't work. So they know that this is a very specific result, and it's not just a side effect of injection or this goggle system that makes everything really bright. So that's coming along, and that's a clinical trial with the FDA, and we'll see if they move to market anytime soon. So that was gene therapy, and that was optogenetics, both gene therapies where you're trying to somehow restore the function that's lost through a gene defect. Critically, both with optogenetics and gene therapy, they really only work if you have a degenerative disease. So if you lost your eye in war, these things aren't going to help. And this is what I was talking about earlier, which is it matters where in your visual system you have the vision loss. So a third approach is called cell therapies, which is kind of a big term. But what you probably heard of is called stem cell therapy. And so that involves, well, you've got cells that died off in your retina. Maybe we can replace those with cells that can replace that function. And so there's been some work on cell therapies. One way is to, some forms of blindness, you lose those photoreceptors I talked about. And so you try to replace those with stem cells. In other forms of blindness, it's actually another part of the retina called the retinal pigment epithelium that's sick. And we try to replace those with stem cells. And so this is just, there's a lot of different forms of blindness or causes of blindness. And this is just addressing some of those. So this is relatively new. Induced pluripotent stem cells really were only discovered in 2007. So that's the ability of us to take a cell that's not a stem cell or is a stem cell that can only become skin or only become hard tissue. And by treating it correctly, you can cause it to move backwards in embryological development so that it could become anything. It becomes pluripotent. And then you have a different process where you treat the cell with different chemicals and environments, and you coax it to become the cell you want it to be, like a photoreceptor or a pigment epithelium cell. And this, like I said, this is very new work. In fact, it was given the Nobel Prize in 2012. So it's very cutting edge. Since 2009, there have been animal trials for stem cell therapies to treat retinitis pigmentosa, Stargardt disease, LCS. And in fact, in 2016, there was a primate study using human stem cells, human embryonic stem cells, to try to restore sight. So we're moving towards human trials. There was a trial way back in 2010 to evaluate safety for embryonic stem cells for retinal pigment epithelium replacement. Now, the problem with stem cells, the reason that it's kind of been around for a while, but it's still stymied and not making it to the market, is that even if we can cause these cells to become the cells that we want them to, they have a hard time connecting into the tissue and wiring up. Because your retina, this tissue that's part of your brain inside your eye, is a network, right? All these cells talk to each other. They have wires that touch each other, and it's important which cells they touch and which cells they don't touch. Well, when a cell dies off and you put another cell in its place, it's hard to make that cell connect to the right partners, but not to the wrong partners. And so that's kind of where we stand, is we're having problems getting those cells to really patch up the tissue that's sick. Another problem with this, which I'll talk about on the next slide, is stem cell therapy, for whatever reason, became this thing that people could do outside of research studies. I have a chat. Let me check this. Stem cells. Did I not define stem cells? So
maybe a little clearer.
Yeah. So stem cells, I'm going to take a step back and define stem cells. So stem cells are, well, it kind of goes back to talking about a human embryo, right? We start as one egg with a sperm fertilizing it, and then you have a single cell. That cell will become all of the cells of a human. So that cell is omnipotent. it can become anything. And then it splits and splits and splits and splits. And then you have a ball of cells. And then that ball of cells starts getting structure. It starts folding and you start getting different shapes. And you'll get a little bud over here that will eventually become the heart. And so when it gets to that point of being a bud that'll become a heart, that omnipotent stem cell has become a pluripotent stem cell. And a pluripotent stem cell is one that can become a lot of different cell types. So your heart has blood vessels, it has muscles, right? It has a lot of different types of cells in it, but it's going to be a heart. It's going to be heart. So it's got some potential, but not all of the potential. And then in adulthood, you and me right now, when we damage our skin heals itself. And that's because there's still stem cells in our skin, and they will divide, and one side will become a skin cell and the other one stays a stem cell. And so those stem cells are our ability to regenerate. And so the idea is we want to have stem cells that will regenerate the cells in the retina. But the retina doesn't really have its own stem cells, so we have to create those. So it's like the stem of a tree. That's where the word comes from. You've got the stem of a tree, which branches and branches and branches and can become all the leaves of the tree.
Thank you.
Sure. So, so there were two cell therapy trials, stem cell trials in 2019 for macular degeneration. I have yet to hear any update on that. I don't think the results have been published yet, but hopefully we'll hear about that soon. But I do want to talk about this issue of rogue stem cell therapy, because I've seen flyers in the Grosse Points for come to the Veterans Memorial and we'll tell you all about stem cells, and you too can get stem cells to cure your macular degeneration. Don't believe them. They're snake oil salesmen. My slideshow seems to have frozen. There we go. So there's a few articles here talking about how the FDA now has the ability to shut down these unapproved use of stem cells. But for a while there, and even now, I think there are clinics that will do stem cell therapy where their sales pitch is awesome. They tell you, look, we're just going to pull some fat out of your butt. And I mean that like out of your gluteus or out of your buttocks, the fat tissue in your butt. They'll pull some of that out. They pull the stem cells out of that. And then they will take your own stem cells, inject them wherever you need to regenerate. And so they're doing this, for example, for arthritis. So they'll inject it into your joints. They say that they can do this for macular degeneration. So they'll inject it in your eye. But importantly, they're doing this without FDA approval. They'll tell you this is an experimental thing. You're taking a risk, but they tend to downplay that risk. And I think they've been really shady about how they've gone about doing it. For example, this, Sunrise Stem Cell Clinic in Florida, a few years ago, they had 3 elderly ladies that had macular degeneration. And they said, hey, we can help you. We're going to take stem cells from your body and inject them back into you. And we think it'll improve your vision. And so they pulled the stem cells out, and then they injected it in the right eye, injected it in the left eye, and sent the ladies home. And all 3 of these women went blind because the stem cell injection did not work well. I think there were some infections, secondary problems. But more importantly, any clinical researcher worth their salt would never inject both eyes at the same time, because with macular degeneration, you still have some functional vision. And by injecting one of the eyes, you're taking a risk of blinding it. So you do them one at a time. Anybody would do that. Any good doctor would do that. But these clinics are working so hard to make money that they're not taking those necessary precautions. So my word of caution is, please, if you think that there's an experimental trial that you want to try out to slow, stop, or restore vision degeneration or restore vision, check with somebody who can tell you whether it's legitimate or not. I'm happy to do so myself. My email is drathbu2@hfhs.org. And I am happy to look at anything you want me to and tell you whether or not it's shady or if it's a legitimate clinical trial. Anyway, that's my soapbox. Don't let people stick needles in your eyes unless you trust them.
Thank you for that.
Sure. It's crazy what's been happening to the unwary. I'm going to skip this slide about cell scaffolds. Um, the next thing I wanted to talk about, so we talked about stem cell therapies, we talked about gene therapy, we've talked about optogenetic therapy. The next one is regeneration. And for a lot of people who have already lost their vision, this is what they're looking at, right? It doesn't matter if you fix my gene, there's no cells left to express that gene, for example. And it's worth mentioning that this is actually, Detroit is a hotbed for this in that Dan Gilbert of, I forget the name now. Anyway, Dan Gilbert, very wealthy man who's bought up half of Detroit, has been funding research in sight restoration through regeneration because he has a family member who's lost vision due to visual degeneration. And so they're looking for a way to regenerate the optic nerve if it's damaged. Quicken Loans is his company. And so the idea here is the optic nerve dies back, and generally with nerves inside the central nervous system, as the optic nerve is, you can't regenerate those cells or those nerves. But they've got some ideas about how to do that. It involves injecting cells and coaxing them to regrow neurons. And so this is very much in early stages, but lots of money has been invested in this. So we'll hopefully see progress soon. I'm going to skip that. And I'm going to skip this study because it was retracted. Okay, so that's kind of the broad field of sight restoration, except for my specialty. And I've got maybe 10 minutes to go over that. This slide, if you can't see it, is a map of the world with many, 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 many labels showing all of the different groups around the world working to restore vision through bionics, through electrical implants. And what's really fascinating and critical about this, something that I think is wonderful about why I'm here at Henry Ford, is every 2 years, Michigan becomes the hub for all of that research in the world when we hold the Eye in the Chip Research Congress, where everybody in the world who's working on restoring vision with electronics comes to Detroit, and we try to move the field forward as a group. So I'm going to skip this, except to say that with electronics, there's a lot of ways that we can implant the electronics into the visual system. For people with retinitis pigmentosa and macular degeneration, the photoreceptors die. And so, but the rest of the eye, the rest of the cells of the eye are healthy. And so if we can wire into the retina of the eye, that network that still exists, and stimulate those cells with electricity, then the optic nerve can take that signal from the retina into the brain, and your brain can do all the processing that it normally does. And so there's a lot of different implants that are retinal implants that are where you have electrodes, an array of electrodes, many dozens or hundreds of electrodes implanted somewhere near the retina, and they stimulate the retina. Other locations that we can have an implant are, I mentioned the brainstem and the thalamus, and also the visual cortex. And one of the more attractive aspects of thalamic or cortical implants is that even if you don't have an eyeball, they should still help you. And that's critical. A lot of people have vision loss due to trauma. And so these gene therapies, these retinal implants, they're not going to help somebody who doesn't have eyes anymore. The only type of visual loss that we're still not anywhere close to solving is if you have damage to your visual cortex. We're many years away from that. But beyond that, we have cortical implants we're developing that may be able to restore sight to almost anybody with vision loss. So retinal implants. They're primarily for diseases where the photoreceptors die, but the rest of the retina is alive. And the primary diseases that we're dealing with there are retinitis pigmentosa, which is about 1 in 4,000 people in the world, and can result from almost over 200 genetic mutations. And macular degeneration, which is also in the world about 1 in 4,000 people, although it's more common here in the US. And it's also the same idea. The photoreceptors die and we put an implant in. Then I have a little pie chart here that talks about the causes of blindness, and it's just a reminder to me to be humble. because worldwide, if we're talking about curing blindness worldwide, there's two important things that we can do. We can make sure that people who need eyeglasses get them, and that'll cure about 40% of the blindness in the world right now. And we can make sure that people with cataracts get the surgeries they need, and that'll cure another third of the blindness that exists. So, if I had gone into this to cure blindness, I would become a cataract surgeon, and I'd travel the world giving cataract surgeries to people who need them. But I think bionics are cool, and that's what I've studied as a scientist. And so for the remaining forms of blindness that we can't treat, bionic vision is a good candidate in most cases. I have a table here, or a list of almost 24 different implants that have been developed over the last 30 years. And 3 of them have actually gotten regulatory approval to be sold to humans and been sold to humans. The first of them is the Alpha AMS device from Germany, the company called Retina Implant. The second is the Argus 2 device from a company called Second Sight, and that was developed in California. And the third is the Iris device developed by a company called Pixium in France, or I should say marketed by them. They actually bought the technology from Germany. So each of those has actually received regulatory approval, at least in Europe, and been sold and implanted in patients. As a result, there are about 500 bionic vision human cyborgs wandering the earth right now. So I've just got a few slides here talking about the Argus. For those of you who can see, there is this array of platinum electrodes in a 6x10 arrangement on a clear silicon surface substrate, and that is implanted on top of the retina. So it goes into the eye, but it is between the retina and the outside and the cornea. So if we look in your eye, we have to look through the implant to see your retina. So this has 60 electrodes, and to understand how much vision it restores, if you take your hand and you make a fist and then put it out at the end of your arm, stick your arm out and hold your fist out there, the amount of the visual world covered by that, it's about 5 or 10 degrees. That's about how big the visual implant is from Argus 2. So it can give you tunnel vision, but none of these devices yet can give you full field vision. And so this device is an array of electrodes that's implanted. It's got some wires that come off of those electrodes. There is a coil that sends information wirelessly between the implant and the external world, just like the newest iPhones, they charge without having to be plugged in. You can put them on a little coil and energy and information is transduced. And then there's a visual processing system. So there's a set of glasses that the patient wears, the goggles, that has a camera in it, and it takes a picture of the visual world, processes that into a signal to be given to those 60 electrodes. That's the Argus. The Alpha AMS is a similar approach, except the implant goes under the retina. So we look through your retina to see the implant. And it also has 1,600 electrodes in a 40x40 array. So many more electrodes, still about the same 5 to 10 degrees of visual field area of vision covered by it. And that got approved in 2013 and again in 2016 in Europe. And that has, so you've got the implant that's under the retina, and then some wires come out of the eye, they go underneath the skin at the side of the head, and then behind the ear, there's a coil under the skin that's implanted and a little circuitry. And then you have a magnetic pad with another coil that you just kind of, it zaps onto the side of your head. So there's nothing that goes through the skin there. And this is the same way that cochlear implants for the deaf work. In fact, the housing for this was cochlear implant housing that was repurposed for bionic vision. And then the third one that was approved and sold to patients is the Iris 2. This one has 150 electrodes. It's very similar to the Argus in that those go on top of the retina. And it has a very similar system in that you have the coil on the side of the eye, and then you have a camera system. Of the 3, the Alpha AMS, the subretinal implant, is the only one that doesn't have a camera system that you have to wear. And that's because the implant that goes under the retina is a photovoltaic array, just like solar cells. So you've got all these little solar cells that take information from the visual world and then directly transduce it to electrical stimulation. So that it really is kind of an implantable Bionic photoreceptor array. So those are the three main competitors. And they have each implanted their devices in many humans. There are results available on YouTube. I have some of those movies here, but I'm running out of time, so I'm not going to be able to show them. But there's been some really impressive results. For those of you who can see, this is an example of those results. I've got 1,600 pixels, and we're going to zoom out a little bit. And you may be able to see that it is a pixelized version of my face. And so there is some sight restoration with these implants. People can see gray levels, and they can see it pixelized. So it's pixelized black and white gray level visual information, little flashing spots of light. What it is good for when it works right, for about half of the implanted patients, they have top level results. For those half of the patients that have good results, they are able to see objects in their environment, navigate around them, detect objects to reach out for, grab them. A few of them with really good results have been able to slowly read letters and read words, kind of like if you were using a big magnifier. But we haven't restored normal sight. So normal vision is 20/20. That's your acuity. That's how good things are. If you're a fighter jet pilot, you may have 20/10, which means you can see at 20 feet what a normal person has to be 10 feet away to see. With these implants, the best that we've seen so far is 2,400. You can see at 20 feet what a normal person can see 400 feet away. So it's still not great. It's still, in many countries, legally blind. And then I've got my demo videos here. But what I wanted to talk about before saying goodbye is, unfortunately, retinitis pigmentosa, I'm going to stop sharing now, just talk to you guys. So unfortunately, retinitis pigmentosa is a rare disease. And it takes a while for people to be blind enough that they qualify for a bionic vision implant. And so the companies that have developed these implants, many of them are actually off the market now. So the Alpha AMS I mentioned was removed from the market in 2019 because the company could not make a profit, even at $100,000 per implant. Second Sight, they've been off the market since the beginning of the pandemic with no plans to come back. And the Iris device from Pixium was pulled from the market only a month or two after it was started being sold. So all three of those that have been approved for market sale are no longer on the market. There is a third, which I think is the next retinal implant to come down the pipeline, called the Prima. And Pixium is also producing that. And the Prima implant is in clinical trials in Europe, and it's in clinical trials in the United States for macular degeneration. And the idea there is that the macular degeneration population is more likely to create enough business for the company to stay in business. There's also a lot of other nice aspects of that implant. It's just a chip. It doesn't have wires connecting it. It's implanted just in the center of vision where vision loss happens, so that you have your natural peripheral vision and then artificial central vision, and you integrate those two. And I'm very eager to see that device come to market in the next few years. And then beyond that, there's a number of cortical implants at the Illinois Institute of Technology. Phil Troik just reported this past year first clinical trials in humans of an implant that honestly, I think the technology was developed by NIH in the 1970s, or at least started. And his team has finally got it to the point where they can put it in humans. And there's 2 or 3 other groups around the world that are developing these cortical implants that go into the visual brain to restore sight. And those are in clinical trials. So we're pretty close, I think, in the next few years to having a few more of these devices brought to market and become available. But it's still going to be a while before the vision that's restored is more than just more useful visual information, before it feels like you got your vision back. What patients report now with any of these treatments, gene therapy, optogenetics, bionic vision, is I can see more than I was able to see before. And if you've lost vision slowly, if you went from sighted to poor vision, maybe you lost your night vision, and then you went to low vision, and then ultra low vision, and eventually blindness, You know that going back a step from ultra low vision to low vision is a big improvement. You know that going from full blindness to ultra low vision is a huge improvement. Even though you can't do much with that vision, any bit that you have, any visual information you have connected to the outside visual world can be very useful. And so it's better to think of these treatments right now as another tool, just like a guide dog or a white cane, that allows you to better deal with the visual world. So yeah, I think that's a good place to close this.
I have a couple questions. Can I throw a couple questions at you?
Please do, please do.
Okay, so I have a few. I've been tracking along through your presentation. And when talking about gene therapy, you mentioned that in one of the procedures, there was an improvement in the non-affected eye. What is the danger of that happening?
Why is that bad?
Yes.
The way gene therapy is supposed to work is we inject the virus particles right next to the cells that we want to infect. They infect those cells, and those cells get healthier, or at least they get restored function. The virus particles shouldn't be going to the other eye. It may be that the virus particles went over to the other eye, and if so, we need to understand that, because that means that these virus particles are moving around more than they're supposed to, and that could be bad for the patient. If they got throughout the rest of the body, you don't need this gene expressed in all of your cells. You don't want your kidney to be light sensitive. I don't know that it hurt you, but it's not intended. And so, when we don't understand something, that's where there can be danger to the patient, or that's a place that there can be danger. Another reason that this might happen is there's a certain component of patient enthusiasm and patient engagement that determines your visual outcome. And I have this problem with my glasses. I tried too hard the last time I got my glasses changed, and the prescription I got works really well if I'm trying really hard. But at the end of the day, when I'm tired, my glasses don't work very well. Because we can influence, our cognition can influence a little bit the evaluation of how good our vision is. So it may be that the patients who got the therapy, and they know they got the therapy, are just trying harder all the time. It could also be that there's some secondary effect. It could be that that improvement is not significant, right? That it's an improvement that's kind of within the boundaries of variability that you would expect. So that's why they're asking for more placebo-controlled tests. Is we've got to figure out why is the eye that should not respond responding.
Oh, okay. That makes sense. What are some of the unique opportunities and challenges specifically around eye research? What makes this maybe a tougher field or makes it more advantageous for research?
I'll go with the good side first. When I tell people that I help make cyborgs and that I'm trying to restore sight to the blind, I don't have to argue about why it's important to provide money for that. A lot of scientists, they do important work, but it's very abstracted. It's very far removed from the patient. And so, they get made fun of in Congress. Why are you doing this weird research on mole rats or whatever? And it is important, but it's a long walk to explain how this very, very niche science is going to help people in the future. I don't have that. So I think that's a good side of doing sight restoration research. I think one of the downsides is it's expensive. It is really expensive. This is a massive undertaking. This is the way I look at my career. And when we have this conference here in Detroit every 2 years, is we are the people in the world. We are the global effort, humanity's effort to cure blindness. And we're putting a lot of money and a lot of effort and a lot of people's life's work into that, because it's a hard problem to solve. We have cochlear implants that restore hearing, but it turns out that the way hearing is encoded at a neural level is a very simple message. And we're able to decode that easily and provide restoration of hearing that people can use so that they can understand speech. And some people even enjoy music with their cochlear implants. Vision is a much more complicated code. And actually, that's where my research at Henry Ford is focused, is understanding that code. I don't develop devices, although we have one idea that we're trying to patent right now. But for the most part, I'm studying that code. So it's a hard problem to solve. It's hard to get money, and with these companies closing, it's become harder because it's harder to get investors because they see that it's not really a good way to make money. But that's not why I'm in this. We're in this to try to cure blindness. So we may have to rethink how we do this and shift towards more of a orphan disease model where the government says, you know what, pharmaceutical companies are not going to put the money in to finish solving this problem. We've got to keep pouring money in as a government, and as nonprofit institutions have to do that from the humanitarian side.
That was actually my next question. Was the cost just seems so prohibitive. So I can see that as an ordinary everyday person, you just would feel that this is so out of reach for you. What ultimately brings costs down? Is it just the size of the population that's in need of the technology, or is it more?
That's part of it. So there's that economy of scale thing going on. So a lot of the aspects of the cost are one-time costs. So whether that solution is good for 1 people or 1 million people, you've already invested that same amount of cost. And it's just the production cost that scales with the number of people. And even that reduces as you have more and more volume. So yeah, it's the more general we can make these implants, the easier it's going to be to make the business case for them. So cortical implants are going to be able to help most people with blindness. If they're blind enough to justify a brain surgery, then they qualify for the implant. And so that may bring the cost down even more. Interestingly, that idea that my group is working on patenting actually could bring the cost of the devices down from 100,000 to 10,000. So there's a lot of work going into trying to reduce the costs. If we were able to charge the same as Luxturna, they're $425,000 per eye, then maybe the companies would have stayed in business. But the companies have been focused on making sure that the people who need these treatments can get them. And with Luxturna, normally you're helping children or young people get sight back for the rest of their life. Whereas with Bionic Vision, often you're helping people who've been blind their whole life get some vision or get some vision restoration. And so the cost benefit analysis is a little bit different there.
I see. I do appreciate the way you framed it as using these as another tool, like, the white tip cane or a leader dog. I think that helps. It certainly helps me put it in perspective and in terms of expectations and things like that. But we will continue to look forward to the research down the road. Is there anything you want to leave people with in terms of parting thoughts for your presentation today?
I would say it's a sense of hope. I've committed my life and I've still got a few years, so I'm going to keep doing this to sight restoration through electronics. There are cyborgs out there already. There's going to be more cyborgs. You're going to meet more of them, and it's going to become more commonplace. But it's going to be a slow road, right? We're going to start off with modest improvements. And as long as we can get the technology to stick and people are using it and working with it, it'll get better and better, just like cochlear implants have. So I'm hopeful. There are a few clinical trials out there. If you're really desperate to try something now, you can contact me and I'll help you find out what clinical trials are running. And there should be something on the market very soon, just a few years, that many people can access. So I think there's hope for sight restoration, not just with electronics, but many of these other approaches. And it's going to be a long time where we have all of these running in parallel. They're kind of competing against each other, but really they're all working towards the same goal.
Well, that's exciting. So thank you for being with us today. I've really enjoyed this conversation.
Yeah, I've enjoyed being here. Let me know when I can come back. Take care.
Thank you.